Okay, everyone. So today what we're going to focus on is a bit of an introduction to the topic of cancer, as that's going to be central to the course project. I'm also going to give you a layout of what constitutes our course project in terms of the writing that's involved, in addition to the specific format that we're going to follow, and also a bit about the deadlines that we have for different components of our course project. So, a little bit of context then. So, let's start off with a discussion about statistics that relate to cancer relevant today. And so, this was taken from this article from 2024. I tried to find something that was more recently published, but more recent data is more country specific as opposed to global. So, this is I would say, the more current. When we're discussing cancer, it is a large complexity of diseases that is characterized by the presence of heterogeneous aberrant cells. And so what I mean by that is going to be a mixture of cells, and that they are not of a normal form. And so we're going to talk a bit about what constitutes that heterogeneity. So these cells have the ability to multiply in healthy tissue in the body and also to spread beyond the tissue of origin, and that's what's known as metastasis. Cancer ranks as a leading cause of global death with a very high burden of incidence. So there were about 20 million new cases in 2022, and also high burden of mortality, approximately 9.7 million deaths in 2022. Now, when we're talking about treatments, and that's going to be a major focus of our course project, we're going to look at how chemical biology has really paved the way to really innovative approaches. But if we're thinking about more layman approaches to treatment, the gold standards tend to be these traditional approaches that have limited effectiveness. So we're talking about surgery, which is effective at early stages, radiation, chemotherapeutics, which are more general systemic approaches that are widely used in different regimens. So we're going to focus a bit on chemotherapeutics, although our course will look beyond just drugs that have been developed for the purposes of the treatment of cancer. So when we're talking about this particular treatment, here we are focused on the use of chemicals to kill rapidly replicating cells in the body via a systemic administration. So that means that either it's an oral administration or something that's delivered through the vein and going through the circulatory system. It remains as one of the major options for the treatment of cancer, and one of the leading family of drugs are the platinum 2 family of compounds. So the idea behind chemotherapy is that you want to deliver a dosage that will maximize the activity of the drug for targeting malignant cells, or at least being able to effectively kill malignant cells while imposing a minimal damage to healthy cells, because given it being a systemic administration, you're not necessarily targeting these cancer cells. But in practice, however, because of that lack of specificity, generally speaking, what you have is a heavy toxicity as a consequence of off-target effects, but also you also have a number of drugs that have a limited window of use, given that the body, or at least tumors, can acquire a resistance against them. So as far as the development of drugs, we're going to talk a bit about the transition from drug development as it used to be for the development of anti-cancer agents to where we stand now. And the beginning of the course, I talked a bit about how chemical biology is also really transcending this field. So the traditional approaches for the production of anti-cancer drugs focus on screening of different families of compounds against model cancer and non-cancer cells. So this type of approach is more of a trial and error. You make compounds or a series of compounds that are related, okay, and then you try to see what activity they have. Do they have the capacity to kill certain types of cancer cells? And so the idea is that you want to see that it does have that effect, but also that it doesn't have the effect of hurting non-cancer cells. So this type of approach is a trial and error empirical approach. And you're looking at the phenotype, so in this case, seeing like what happens to the cell's survival or the health of the cell as an indicator of the effectiveness. So there have been some formalized structure activity relationship studies where you design a compound with a particular structure with the intent of having an end goal. But this type of approach still is more of an empirical nature. But this has changed dramatically over the years, and so today we are aware of many different technologies that are being used. But I would say, as far as the field itself, this transition has been to some extent slow. So, what have led some of these changes? Of the 210 new molecular entities, or referred to as NMEs, approved by the FDA from 2010 to 2016, the funding from the National Institute of Health has centered on basic research, and that funding has contributed enormously to the development of these new molecular entities. The work focused on the biological targets as opposed to developing drugs and testing them. So here we see the transition from an empirical approach to an approach that is target based. Part of this is led by people really, giving their insight as to what should be the direction of drug development. So someone like Dr. Robert Weinberg, who is a pioneer in the cancer biology field, um, believing that a landmark anti-cancer drug or treatment would be one that targets molecular mechanisms that keep cancer cells alive, and more than one molecular mechanism, so that you have a synergy of approaches to basically trigger a number of ways that could attack cancer cells. So over the years, people have really dived into what constitutes cancer. And I've already mentioned that cancer is not one type of disease in terms of how it manifests in different tissue and different organs. But there are some commonalities, and these have been identified by people over the years, which we're going to refer to them as the hallmarks of cancer. So in 2011, this article was published, Hallmarks of Cancer, the Next Generation, which helped to identify what are these hallmarks. Here we have 8 distinctive and complementary capabilities acquired through the multi-step development of human tumors that enable growth and metastatic dissemination. So such as sustained proliferative signaling, so that capacity to be able to thrive and proliferate, evade growth suppressors, so evade anything that's going to suppress the ability to continue replicating, resistant cell death. So there are natural cell death pathways that are triggered when a cell has gone through its cycle and needs to then, reach the point where it's no longer healthy and has to experience death. Well, cancer has learned to be able to avoid those type of pathways, enabling replicative immortality, inducing angiogenesis, which is essentially the formation of new blood vessels, and that provides nutrients that allows for the potential movement of a tumor cell from one location, primary location, to another. So that leads into then activating invasion and metastasis, excuse me, reprogramming cellular metabolism. So you see the abnormality of cancer cells being able to thrive with certain nutrients that a normal cell may not have the capability of doing so, given that the tumor environment can be quite, heterogeneous. And so you don't have necessarily the same supply of resources that a normal cell will have. Also, avoiding the immune destruction, as our immune system has the capability of targeting things that are foreign to the body and being able to attack them. But additional features have been identified over the years. In that same article, these were additional features that were identified. For instance, these 2 enabling processes, the genome instability, so there being a high rate of DNA mutations and other genetic changes. Also, tumor promoting inflammation, so inflammatory response that stimulates cell proliferation. And so you see in this perspective here, the different hallmarks of cancer that I have, to some extent have introduced, and so how they lead into what allows for cancer to prosper and to spread. Other additional features, and so this was published in the Hallmarks of Cancer New Dimension in 2022 article that have emerged. For instance, polymorphic microbiome is a complex community of microbes that can be directly carcinogenic. They impact the immune system and affect how well cancer therapies work, keeping in mind that your cancer cells can be mixed with its own microbiome, and that feeds into how resistant the cancer can be. Senescent cells, which are cells that stop dividing but do not die. Phenotypic plasticity is the ability of an organism to change its phenotype in response to environmental conditions. Also, non-mutational epigenetic reprogramming, which refers to a process where gene expression patterns change significantly without any alteration in the DNA sequence itself, essentially meaning the function of genes is altered through epigenetic mechanisms like DNA methylation. Okay, so that is just a change in a functional group, let's say somewhere in the nucleotides, okay, or a histone modification of the chromosomes rather than an actual genetic mutation. So here again is the larger perspective of the hallmarks of cancer. And so that being published in that 2022 article. But there are additional ones to keep in mind. And this has to do with, what's surrounding the structure of tumors. In addition to what are other factors that bleed into the biomolecular pathways that enable cancer to thrive. So you have the feature of the enhanced permeability and retention effect, and that is that the vasculature that surrounds tumor cells tends to be quite leaky, and as a consequence of that, they lack an effective lymphatic drainage. And so that is often an important feature that people try to take advantage of for drug delivery. For instance, the use of nanoparticles, where you can attach different drugs onto the nanoparticle, which is basically serving as a vehicle for delivery. And so these nanoparticles can penetrate close to the environment where the tumor cell is. And because of this limited drainage, they can exist at a longer time frame in the presence of the tumor cells, enough for there to be a higher uptake and also a drug payload as far as what's being delivered into cells. In addition, you have a number of metals that are essential, and because of how prevalent they are in cancer cells, and also because of the importance that they play in different biochemical pathways, they can further enable cancer to thrive. Here's a representation of how cancer can depend on iron. This is the perspective of, and now I put this as iron 3, but probably I should have just left it as iron in general, that what you see in a healthy cell is this really nice balance between how much iron gets in and how much iron is actually utilized, because most cells have a controlled level of iron. Well, for cancer cells, what you have is a different balance in this iron. You have a lot of uptake going into the cancer cell because of a higher utility of this iron. So what you see is a lot of expression of proteins for the uptake of the iron and the expression of proteins that are responsible for, activating this iron for different purposes to keep cells alive. So just some key notes: higher expression of the transfer receptor, and that's the receptor that captures iron from the protein transfer and brings iron in. Higher expression of the enzyme ribonucleotide reductase, which is an iron-based enzyme, which is responsible for the um, the building blocks, so nucleotides, the transmission of nucleotides, to enable DNA production and also, consequence of this, greater proliferation. It also participates in DNA repair. So let's talk a bit about this idea of the heterogeneous microtumor environment. So here is a cartoon representation of the constitution of, let's say, a solid tumor, and it consists of some cancer stem cells, some, I guess we would call them more regular cells, in addition to having other types of cells that are, participating in the structure. So that is to show us that what we have is an assembly of distinct cell types. And as a consequence of that, when you're thinking about a drug design, drugs will not necessarily have the same impact in all of these cell types. So in addition to this, is to think about the composition of the tumor as what is actually supporting it. So what we have to think about is the extracellular matrix, so obviously outside of the cellular environment, how that contributes to the formation of this tumor. And so in addition to the different population of cells, you also have a different population of nutrients and resources that are accessible to the different cells. What contributes to that is the presence of different blood vessels that may be in close proximity to some of the cells and not so close proximity to others. So what results come from that? Well, depending on how close you are to these blood vessels, you may have a higher access to oxygen, or if you're further away, a much lower access. So you're going to have this sort of gradient of oxygen the further away you are. In addition, you're going to have some changes in pH the further away you are from the blood. Blood being something that's buffered, the further away you are from something that's buffered, that tends to lead to a more of an acidic environment. All right, so regions far away from the blood supply are hypoxic. That means that they're low in oxygen, so they tend to survive under different conditions that perhaps if you design a drug that is oxygen dependent, it may not be something that will be effective in an environment that can thrive in a low oxygen amount. You also have invasive phenotype may undergo a epithelial mesenchymal transition or EMT, as that's depicted here, which essentially results in a migration into neighboring blood vessels that can then lead to metastasis. So here, what I'm showing you is different approaches that people over the years have tried to invoke in order to target the different hallmarks of cancer. So some of these targets, or at least some of these strategies, constitutes some of the approaches or innovation that we're going to potentially focus on in our course project. All right, so what I wanted to talk a bit about is the more traditional approaches of how drug development goes from the laboratory environment and can ultimately lead into a testing into living organisms. So the typical approach is to start first with, let's say that you make your compounds, you've identified them, and then you're interested in testing them, okay? And whether you made this in an empirical fashion or you actually included structures that are known to have a certain activity against cancer cells, okay? Depending on the approach, this is a typical methodology, which is that you start to use cells as your benchmark for evaluating activity. So what are we looking at here? So we're looking at then the approaches of assaying how cells respond to your actual compound. So you either design an anti-proliferative study where you're looking at whether your compounds are inhibiting the growth of the cells. You can go into further types of cytotoxicity, which can be just something that's strictly anti-proliferative, or looking at whether a drug can actually induce the death of cells. So when you're doing these type of experiments, you have to grow up your cells, and they are living things, obviously outside. When we talk about mammalian cells, they're outside of a living organism, but they're still living things um, that you have to maintain under optimal conditions for them to be able to grow. So a typical approach of culturing cells is in an incubator that maintains a controlled temperature, say 37 degrees, and a humidified atmosphere, also conditions that are going to enable your solution to be buffered. So typically you have this incubator flowing with 5% carbon dioxide. Carbon dioxide helps to, when it mixes with your solutions, helps to maintain a certain amount of carbonate that is going to buffer your media in which your cells grow up. So the typical cell culture media consists of a number of nutrients that enable cells to survive. So what it tries to approximate is the fluid portion of blood. So you have salts, you have certain common acids, vitamins, glucose, which is a big source of energy. You have glutathione, which is a biological reducing agent and participates in many of the control, the regulation of redox processes in the body, additives. And so you can supplement your media with with a source of serum because the typical blood consists of serum or consists of protein nutrients. And so oftentimes these media will get supplemented with a bottle of fetal bovine serum. So this is blood from cows. And that is to supply the essential proteins that are appropriate for growth. In addition, you have some media that is supplemented with antibiotics because you want to avoid contamination from known microbes, known bacteria. And oftentimes we will include a pH sensitive dye, and that dye, and in this case phenol red, which makes this particular bottle red, is an indicator to let you know if your media is in the appropriate conditions or if you're having a loss of pH control. Another really important thing is to consider the solubility of your drugs. So oftentimes drugs are not immediately soluble in water, but can be made soluble. So you oftentimes have to first solubilize your drug into a different solvent that is biocompatible, also that is miscible with water. And there you have to keep in mind, is the drug something that is stable in this other solvent? So here's an article about some platinum compounds that cannot be dissolved in DMSO. That's considered a universal solvent, But a problem with that particular solvent for those compounds is that it actually changes their structure. So you put that in that solvent, you're already forming something completely different. And then you mix it with water, you're not sure what exactly you're actually studying. So that's something to keep in mind. Solubility of compound, but what solvents keep your compounds stable, and that can be used for these type of assays. So solubility is key, whether or not a compound can even be used in terms of testing against cells. So a typical assay for probing the activity of compounds tends to be something that can be a high throughput. So you can do multiple experiments and get a lot of data as quickly as possible. An example would be an experiment that's designed where you can run 96 experiments. And so here's an example of a 96 well plate where you can do an experiment in each individual well. And so what tends to happen is that you will place your cells into each of these wells, and there may be some controls where you don't want to actually place the cells, you just want to look at the background of whatever conditions you're monitoring. So you put the cells, and so depending on the type of cells that you're working with, you want for them to equilibrate in this environment for a certain amount of time. So here then the type of cells that you're working with will matter. So there are cells that are adherent that must stick to the surface of the actual plastic in order for you to probe the activity of a compound. Those typically require at least 12 hours to equilibrate, maybe a whole day. Whereas there are cells that are non-adherent, so they don't require that sticking on to the actual surface, but it's recommended to still have some amount of time of incubation so that you know that all the cells after you have plated them have equilibrated. So after that time of equilibration, you would treat the cells with compounds and you would do a dose response. You would vary different concentrations with relevant controls. So controls are compounds that have known activity so that you can test whether or not your assay is working well because your controls are behaving in the way that you would expect. So people will typically run these type of tests from 1 to 3 days. The typical recommendation is more time just because certain compounds take some amount of time to become activated. And then because of that lag phase, you may not see an effect that is very clear or at least optimal in a short amount of time. Then often a dye is used to quantify the number of living cells in treated or versus uncontrolled conditions. And so I want to be a little careful, not necessarily are you looking at whether or not cell death is occurring, but you're looking at cells that are alive, that are viable. And so that's the relative count, is what's happening to the amount of the viable cells. The National Cancer Institute, which is an institute of the National Institutes of Health, recommends measuring the number of cells after the 1 day incubation. So this will be the time zero number of cells. You've waited that amount of time of incubation before you treated the compound. How many cells do you have there? So a typical dye would be this type of dye, which is a tetrazolium salt. So this is it could be something like MTT or MTS. What I have depicted here is MTT, that's just an abbreviation. So the idea is that what you're going to do is you assay the cells in the presence of your compounds, and then after whatever amount of time, 1 to 3 days, you then will place this dye into the media where the cells are located, and then the cells that are alive, that are viable, will uptake this compound. And because of the presence of mitochondrial reductases, this MTT will become reduced and it will form into what is referred to as a formazine product. So it is bioreduced by nicotinamide adenine dinucleotide, so the NADH, with the help of hydrogenases. You get the formation of this product, and what's distinctive is that this product is purple. So this is essentially a colorimetric assay, absorbance at 595. And so based on the intensity of the absorbance, you can correlate that to the relative number of viable cells, or at least the relative percentage of viable cells. So here's just a visual representation of a dose response assay where apparently you're going from high concentration to low concentration because what we're seeing is an increase in the coloration of the product. The more intense the color, the more viable cells you have. The lower the color, the less viable cells you have. So higher concentration should lead to less viable cells. Lower concentration should lead to a darker purple color. So what type of data do we expect to see? So here is a representation of the process data. where we're looking at cell viability, and this actually should be percent cell viability, so the percentage got cut off. And so what we're looking at is the maximum color will represent the maximum amount of cells, that will be your 100%, to then no color, which would be 0%, and that's an indication of there being no viable cells. Okay. So what we're going to see in a ideal experiment is that you're going to look at the responses of the cells to the different concentration. You will see that as you increase the concentration, you will have this behavior where the viability decreases from 100% down to 0%. But the reality is not always like this. So again, 100% represents the maximum number of cells present in the no compound control upon completion of the experiment, or it can represent that you've had that maintained that maximum even in the presence of the compound, but the compound's concentration was so low that it had no effect. The 0% represents a complete loss of living cells. But if you're doing this experiment where you're basing it off of the measurement at the initial time point, then what this indicates is that it indicates that no further cell growth from the initial point occurred, even in the presence of the compounds. All right, so what is an important way of analyzing this data? So we have this curve, and I've plotted percent viability versus the log concentration of the compound. So the parameter that we want to look at is this IC50 parameter. That is the inhibitory concentration at which 50% of the maximum number of cells are viable. That is indicative of a compound's antiproliferative effect, meaning that that is the concentration at which that compound basically inhibits 50% of the total growth. And so that's the parameter that we measure. So this way of doing the experiment would be an antiproliferative study. So in reality, though, you don't necessarily get all the way down to 0%. Okay, so you may have a cutoff that's 20%, 24%, because certain compounds, after a certain point, may just not exhibit much of an activity. So oftentimes what people measure is the maximum signal to the minimum signal. And so that becomes a measure of the relative IC50 if you are measuring just the absolute color change. So that becomes the inhibitory concentration at which 50% of the signal between maximum number of cells are viable and minimal number of cells are observed. If you're measuring with respect to the initial time point, then you may not get back down to that initial time point number. You might get something that's higher than that, and still it will be a relative IC50 because it's from the maximum signal to that minimum. That's not the actual amount of cells that you initially had. So if the number of cells for day one incubation of cells is actually measured, okay, then another way that you can do this experiment to get some idea about the ability of the compound to induce cell death is by measuring the LC50. So let's see what that means. That's the lethal concentration at which you have killed 50% of the cells. All right, so how do we get to that point? Well, 100% in this context refers to the number of cells present at the initial time point, where 0% refers to complete loss of all those cells, okay? So in this case then, the LC50, the lethality concentration at which there is 50% of the day one cell number, that means that that is the concentration at which you have killed 50% of those cells that you had initially. And so this approach will give you a measure of the actual cytotoxic cell death capability of a given compound. So now we have to think about what type of IC50 values we want, and also measuring the activity of this compound against different cells, cancerous and non-cancerous. So there are two desirable properties. So it'll be what type of IC50 values we really want, and also a therapeutic index, which is a measure of the selectivity of that compound versus cancer cells. The general rule of thumb is that you want to aim for IC50 values that are low micromolar or even lower than that, nanomolar. And that's for practical purposes. So what do I mean by that? Well, we have to think about the method of administration. So let's imagine that we're trying to introduce this intravenously. So you're taking a solution that you've prepared with your compound, you're putting into your veins. It's going to flow through the circulatory system. That's going to dilute your compound. So keep in mind that the IC50, that's the concentration where you see an effect of the compound. So basically, you don't want that dilution to be so significant that that compound becomes so dilute, it doesn't even show any potency against when it reaches the tumor environment, that it doesn't show any activity. So if a compound has an IC50 in the mid micromolar range, then you would need to prepare fairly high concentrations of that compound so that when you introduce it into your blood system, you're able to dilute it to a concentration range that it has an effect. So the typical adult human has 5 liters of blood. So if you administer a drug, let's say via IV, and you can administer like 50 milliliters or even a liter, then you have to think about how that volume will get diluted by the blood, and so how much that concentration will get lowered. So keeping in mind this math, let's say 50 milliliters administration, you're putting into 5 liters of blood. So roughly, if you're going from 5 millimolar, um, that will give you 50 micromolar. That's basically the IC50. You see how much of a high concentration you have to make of a compound. And many compounds don't have solubility of that amount. Also, as I mentioned to you, is the therapeutic index, and that's a measure of how effective the compound is against the model cancer cell lines versus the model non-cancer cell lines. So a parameter you want to see is the following, where you have the IC50 against the non-cancer cell lines being really high. The higher it is, the less effective it is. So you want that to be much higher than it is for the IC50 against the cancer cell, such that the ratio is something much, much greater than 1. Okay, so these are the desirable parameters. A low IC50 value, something lower than a micromolar, and a high therapeutic index. Well, one thing to be cautious of is that a lot of these studies are just focused on one cell type, okay? And I already mentioned to you that a lot of the cancers are very, very heterogeneous. So you have to think about the limitations of an experiment that's only measuring the IC50 against one particular cell line. So here's an interesting story where we're looking at these ruthenium compounds, where what was studied initially was the effect of these compounds against a primary tumor. And as it turned out, the effect against the primary tumors was actually quite weak. But when those compounds were screened against a secondary metastasis tumor that came from this primary tumor, there, these compounds showed a really potent activity, so really low IC50 values. So a bit of a take-home message there is that like you should focus on the effect on several cell lines, where because the reality is that looking at just one cell line does not give a full picture of how effective that compound can be across different cells of that particular cancer type. Also, it's very important that you do try to follow a more target-based drug discovery approach. So you want to design your compounds to have a particular structural feature that will be able to reach a target of interest. Things that people do to get a sense of how effective a compound may be is studying its ability to cross the cell membrane, to be uptaken. So it can be something that's passive, that because of the size of the compound, it can be inserted into the cell, or it can be something that has a structure or some feature of the structure that can be recognized by a receptor that can bring in the compound. So oftentimes people are interested in that because you want to optimize how much of the drug you can get in. And so you can try to study how much uptake there is by doing some elemental analysis. So it could be an analytical tool such as atomic absorption spectroscopy that can measure low levels of elements, but preferably it can be something like ICP-MS, which is inductively coupled plasma mass spectrometry, which can measure things down to the parts per billion level. It can even be parts per trillion to some extent. So the information you can get here is the amount of element that's going into per cell. But this process can be cumbersome, and especially depending on what type of compounds you have and how many you have. Other ways that you can try to get this measure and not as cumbersome can be such as having an imaging agent. So we talked a little bit earlier in the journal club, the use of ratiometric probes that can provide quantitative measurements, at least relative quantitative measurements, of how much of something is being bound to a molecule or metal of interest versus the free form of the probe. So this can be a direct intracellular measurement as long as the probe exists. Okay, so it requires that the probe actually has to exist, or maybe you have to create it yourself. Then you can use this information to do uptake plots where you measure uptake over time. Okay, so it's not something that's going to necessarily be instantaneous. That's why you typically do experiments over the course of 3 days. So you set a time frame of how much time do you want to look at the uptake of something, because typically over a certain amount of time, you're going to get an equilibrium where you're not going to have any necessarily additional uptake. So you measure uptake over time, and it could indicate whether a route of uptake is passive diffusion or transporter assisted, but it's not necessarily straightforward to analyze. But also important to note that increased uptake does not necessarily equate to greater potency. As I've mentioned to you, when you do some dose dependent studies, there are times where higher dosages does not actually increase the effectiveness of the compound, that you reach the limit of how effective the compound can be. Obviously, there are limitations with cell work. As I've described to you, you're basically doing one cell at a time. And so you're not invoking the complexity of how cells in tumors actually grow. Also, the type of cell assays are the cells that you're working with, but these are commercially produced cells. So they're commercially produced and manufactured in such a way to make these cells quite resilient to manipulation in the laboratory. So their genetic makeup has been altered to respond to the environment of the culture media and also the culturing environment or the process. So a way to try to overcome this limitation, so some people do this, is to work with a three-dimensional cell culturing approach. So if you're thinking about, replicating something that is a solid tumor, what you can try to do is grow your cells in a spheroid, which has a three-dimensional structure, so that you can get the that heterogeneous environment where as you go from the surface to the more inside the actual spheroid, you're going to get the gradients of changes in the amount of oxygen, changes in pH, changes in the amount of nutrients. So then you can see whether your compound can affect that particular cell line, right? And under what condition can it affect, like just more of the superficial part, can it affect the whole thing? Of course, still limitations because you're still working with one cell type, but you're getting a three-dimensional structure that's helping get closer to the reality. So once you complete cell studies and then you identify compounds that reach the desired benchmark of IC50, reach the desired benchmark of selectivity, then people will move on to animal studies. And so I'm going to be very brief here, okay, because this could be a lecture in itself. An example of an animal study could be a patient-derived tumor xenograft. Okay, so what does that mean? Well, you can take tumors from a human cancer patient, you take them, and you can implant them into mice. And so basically here is a way to do sort of try to get towards personalized medication. So those tumors that have been planted can be grown for several generations in these host mice. And so the live cancer patient cells, in addition to this tumor genetic data, can be given to the investigator. So you have not just the information regarding the patient, but you also have genomic information. And so this can help perform patient-specific drug studies, where you're basically testing different compounds and seeing how this implanted tumor responds. The disadvantage is that the work must be done under immune compromised conditions, as you're putting human material into a rodent, and so that's going to be foreign to the rodent. Other type of animal studies can be where it's syngenetic models where tumor implants derived from the same animal. So you take uh, a mouse tumor and you put it into the same type of mice. So these type of experiments are not immune compromised because now you have the immune system of the mouse being able to respond in addition to how the drug is, affecting the tumor of interest. The disadvantage is that you're looking at a type of tumor that is not human derived, right? So again, focusing on animal studies can be its own series of lectures, but just to give us an overview of what you can do for further testing. All right, so I'm going to stop it there because I already discussed in the first lecture the process of translating from preclinical to clinical trial. So I want you to review that information to give you a perspective of how then from the successes of the preclinical, what will lead into testing things for clinical trials. So I don't want to repeat that. What I want to do now is to focus on what's going to be the course project. So essentially, collectively, we're going to write a perspective review, but each of us individually will write a section of it. And so what I want to talk about is a bit about how you go and develop a manuscript that's going to be ultimately produced for peer review and dissemination. So here is going to be critical to be familiar with the literature. So the main goal of this course is to foster new different analytical tools in the biochemical biology field that have been designed to get a better understanding of biology, also of chemistry, to be able to answer some really important societal questions. And so we're going basically to take this type of strategy, but focusing on developments, recent developments for the treatment of cancer. And it does not necessarily have to be strategies for specifically treating cancer, it can also be for diagnosing cancer. This will require you to become familiar with the literature. So through the course, you're going to be given lectures from specialists in the field, some that are cancer specialists. And so the idea is that you read their work to become more knowledgeable about what's actually occurring today. So you become immersed in literature, both the primary, which is hands-on research, and the secondary, which is research that's based on data that's already available. And that's going to help you to then try to focus on what's going to be your actual central objective. To help you develop your story, we're going to look at how we can prepare the drafts and visuals. So the deadlines that we have for this project are going to guide you towards those goals of creating your drafts, of thinking about the visuals you're going to use to represent key ideas. Another thing that then this will be more towards my end. What I'm going to do as far as this work is select the journal that this particular article will be suitable for and translate it into the format for submission into that journal. After that point, we are going to then experience what is the peer review process. That is, that you're going to have, as we discussed earlier in the course, peers in the field that we're not collaborating with, who are going to assess the work. They should be specialists in the field, and they will see what things are valuable about the work to recommend its publication or to recommend if more things need to be added, changes need to be done. So based on the peer review, we will revise. So this will be after the course, I will, stay in contact with you so we can see what we can do to try to get this into its final form. We will attend to the revisions and see if further peer review is necessary. What is the response? Or is it approved for publication? And obviously, we're doing this to have a purpose, not just to just have a product and then be done with it, but also to be able to disseminate it. So the key thing is to try to, once this article is published, is to participate in the dissemination promotion part of the work. Okay, that will be a critical component. So we're working on individual contributions to what will be a perspective review. So what is a perspective review? It is a type of investigation in which you critically analyze contributions to a particular field. It elucidates the problem that different research teams are tackling, are not meant to be a laundry grocery list reporting of findings. So here you're going to focus on a particular topic and find articles that are immersed in that topic. So you're going to be focused on a certain published work. It's not going to be something that's very general. You give it insight into the current status of the field. You highlight the major advances of the field. And so depending on the topic, we're going to talk about that in a moment. And as part of this, you should include older literature that sets the field, okay, but also current literature, where the field is going. It serves to highlight the significance of the field and to identify new research directions for the field. And the new research directions will be something that comes from you. From your assessment of the work, where can we go next with this information? So how do you tackle the writing of a perspective article? Well, very important to become familiar with the key players. Who are the people who are moving this particular area, this particular field? That requires you to read and read some more. Critically assess the experimental approaches taken and identify accomplishments and where applicable limitations. So there will be limitations of different approaches. The key thing is to highlight them, see how the field in that particular area can revise things to improve on these limitations. One thing is not to attack people, not to offend them, not to say that this will never work, make suggestions, or think about ways that you can see what valuable things have been obtained through this particular set of studies. So what I suggest is search the website of the key players. Oftentimes, these are faculty universities that provide extensive information about what they're actually doing that goes beyond just the articles that you have read. That helps you to contextualize the work that they're doing. It also personalizes the work and also the environment in which the work is being done. What tools do they have available to be able to pull off the work that they're doing? When working with others, it's good to compare notes. So this is clearly an individual project, but we will be assembling things together. So as you see, we'll have a moment where everyone will present. So it's very good to give each other feedback, and so we can be our own peer review. How to decide what literature to focus on. So to an extent, I'm going to guide you with that. So outline what will be the topics of interest for your perspective review. A perspective review is meant to be a very focused study and not general. Identify the key topics that you can effectively analyze and that you can see something that is unique or adds a refreshing spin to the commentary. Gather a collection of articles of the topic of interest. So you would say that I give you a certain number to focus on. You might want to go beyond that because maybe the minimum that I gave you and you've only searched for the minimum and not and a few of them are good, you're not going to have enough material. So try to go for more. You prepare an annotated bibliography. So we'll look at this annotated bibliography. You focus on primary references. Only refer to previous review articles to help you contextualize work, but do not replicate the review. So I'm actually going to give you a little bit later the format for annotated bibliography. I'm not going to discuss it here. I'll just give you the instructions for this. Outline what will be the topics of interest for your perspective review. So do not cite a paper that mentions a point that is not the main focus of the manuscript. I see that often, where there's a sentence that came from an article, and so people will reference that article, but that sentence is actually a minor part of the whole article. So the article focuses on something totally different. So that would not be an appropriate reference. So here's an example. Let's say you're reading a paper about gold chemistry, and the authors briefly mentioned that the Chinese used gold therapeutically thousands of years ago. This article would not serve as a good reference for the discussion of gold in traditional Chinese medicine if basically that's just one sentence that talks about the historical use in China. All right, so find an article that does indeed focus on traditional Chinese medicine. That's going to be the focus of what you're going to do. Obviously, that's not going to be for this particular assignment. So instead, search for an article that expands on the point of interest. Prepare your library of references. Use software like EndNote to keep your library preserved. So, how to incorporate visual. And for me, storytelling is one of the key things of whatever you write. So, the effectiveness of how you tell your story is also conveyed through the visuals you include. So, images say more than words and help to effectively tell your story. I often recommend you to prepare your own figures. So there are different programs that you can use there. There's artificial intelligence, programs that have artificial intelligence that help you design your figures. You can use programs such as ChemDraw for your chemical structures. You can even use PowerPoint for help to design the layout of your images. Adobe Illustrator, not so user-friendly, but quite helpful. PyMol, BioRender. I have access to BioRender. I can give you access to my version so that you can create visuals for our work. Prepare tables where necessary. So maybe you want to collate data from different articles and to create your own tables. So you're basically assessing data that exists, but in a new perspective. Keep in mind that a review is your own investigation, and you can present information in ways it has not been previously done so. If you want to use published figures, then that's perfectly fine. You must obtain copyright permission, and I can show you how to obtain that. So some figures require payment for use. And so if that happens, it needs to be pretty cheap. So don't worry about it. I will supply funds if you want a figure that seems to be quite useful for the manuscript, and we can create something that's our own. We can go this route, but again, we have to be conservative about the funds. So journals often have templates that you must use to construct the final form of your manuscript. Here, we're going to just create a traditional Word document, but when we go from your contributions to me assembling all of them, I will transition into the format of the journal. So you prepare the manuscript in the order required by the journal. There are often page character limits and limits in the number of figures and tables. You prepare a captivating title. That's true no matter what the journal or format is. Effective storytelling starts with the title. So I often see people write class report or research assignment. And that just shows a lack of creativity. It shows that a disinterest in the work. Write a compelling abstract to start off the manuscript. The abstract should be concise but informative, and don't make it a laundry list. Don't make it a grocery list of things. Then you establish the focus of your review in the introduction. So I'm going to give you a topic as far as the overview topic. You make a selection of what specific you want to focus on, and then you establish that focus in the introduction. You contextualize the significance of the review's subject matter. You identify the key topics that you're going to elaborate on based on the primary articles that you're going to focus on. And you briefly present what new insight you will be providing in this introduction. Then you effectively layer your story in the discussion and conclusion sections. You arrange your story in a coherent collection of topics. You present data and analysis. You highlight key accomplishments and limitations and describe how they further the field. Subsequent sections should build on from the previous one. So you don't want it to be a layer where you present the topic and then you present papers that have no connectivity. See this as a more holistic storytelling approach. Conclude with an overview of all topics discussed while focusing on your collective vision and what research avenues need further exploration. And your acknowledgements. Recognize any source of funding that provides you research funds because perhaps you're going to take additional time than usual. And some of you do have some research funds that help you to, devote this time to focus on the work. So if you're using extra time, and this cuts into what would be research time, then I suggest you should acknowledge these sources of funds. Also recognize people that have provided you with direct input or feedback. So if there's anyone that's giving you suggestions as to what you should focus on as far as studying, what references, or give you some actual really useful data, you should acknowledge them. Okay, so how to submit for peer review. So this actually is beyond what you're going to focus on. It's me having the collective work, and I want to submit it to the journal. For every submission, you always prepare a cover letter. So I'm teaching you this because I want you to be aware of this process of peer review. We've already talked to some degree about peer review. A major opportunity is to convince the editor of the importance of your work through the letter. Where appropriate, you suggest an editor from the editorial board that is suitable to evaluate your work and help it to move through peer review and help to improve upon it. And then you also recommend at least three referees, three external experts that you do not collaborate, they cannot be friends, to properly examine your work. So these referees will give the editor the feedback as to whether or not the manuscript is worthy of publication. Prepare an interesting but simple table of contents or graphical abstract. Okay, so these visuals, often journals will require it, and they tend to be really small, so you need to be really focused on what you want to show in that visual to try to capture at least one big idea. Do not overstuff the figure because it's meant to be small and concise. Okay, now let's go focus on the specific topic at hand. So I want us to work on chemical biology breakthroughs in the treatment of cancer. But then we can probably soften that word treatment because we may want to go beyond just like strategies that directly treat cancer. So this overview. Throughout this course, you're being introduced to the foundational principles and strategies of chemical biology as applied to contemporary biomedical research. The course examines how chemical tools are used to interrogate complex biochemical environments and apply recently acquired insight to address real-world research questions. For the course project, we will direct our attention to groundbreaking advances and technological innovations that are paving the way to the development of long-term and highly impactful cancer treatments. Through cancer-focused case studies and primary literature, we will construct a review article in which each of you will focus on a specific innovation. Again, this will be an individual project that will later become a group project. Below, you will find a list of possible innovations which to choose from. You also have the option to go outside of the list by searching for a recent breakthrough discovery from the last 5 years that is revolutionizing the field. So here is a possible innovations that you can choose from. And so I'm going to try to make sure that we have a mixture of these, not just people choosing the same ones. Okay, so I would say that we should probably do this based on a list of preferences. So I would like you to do that. Or you can choose your own. So you go outside of the list. And so if you choose your outside and you justify it, then that will be your topic to focus on. Right. But if you choose to focus on this list, please provide me your list of preferences. So how are you gonna do your list of preferences? I think there are 12 here. You're gonna rank them from most interested in to the least. So 1 would be the most interested, 12 would be the least interested. So what's the format? You are to write a 10-page manuscript in which you will focus on a specific innovation and dissect 3 recent primary articles from within the last 5 years in which that innovation has been applied. To provide a well-rounded discussion, it's important that the articles selected come from different research teams where it's actually possible, because you don't want just it coming from one team and we're not seeing much difference in thought or application. The following is a suggested breakdown of the writing, where you start with the innovation. That would be one of these innovations. You give the context, write a brief history of the innovation and a timeline of developments. Explain how, and I would say timeline as an actual visual timeline. Explain how the innovation works and what tools are used in its implementation. Then the body, the main part, will be the dissection of the article, 8 pages. Probably I would say maybe this will be more like 7.5 to leave room for your conclusions. You introduce each article by focusing on the big picture scope, not the actual article itself. So you want to avoid introducing each paper by its name, instead focus on the particular application. Try to transition between discussion of each article by emphasizing the progression in the use of the innovation to tackle another challenge in the field. Discuss key experiments performed and provide concrete data and your own assessment of the data. Include quantitative results, and I'm very particular about this. I see a lot of qualitative discussions that don't actually say anything concrete. So avoid being overly general about describing differences. Do not say something is more effective than something else, because more effective is there's no gauge, there's no benchmark. So without providing a benchmark for comparison, include figures. We already talked a little about this, the importance of visuals. Where possible, create your own figures. You're also welcome to use figures from the article as long as the figures are of high resolution and have no fees or minimal fees if we intend to publish the work. Conclusions. So here, this should be no more than half a page. Review the success and highlights of the application of the innovation and the possibility for real-life use. Because some of these are, very promising, but will they actually reach the market? Or maybe you won't know that answer, but you can give some suggestions. Discuss possible challenges that could be tackled with the innovation and any still inherent limitations of that. So please write this in Arial, 12-point font, double space with 1-inch margin. So that's pretty, as far as the amount that I'm explaining, is not a significant amount of work. Okay, so these are your corresponding deadlines. So February 17th to selection of the topic, March 3rd to prepare your annotated bibliography on 3 pertinent articles. So that's the minimum. I would suggest more. So the annotated bibliography should include a 2-paragraph description of the objective, methodology, and accomplishments of the work, and should be progressive in that you define how the articles relate to one another and build on key ideas. So again, I will supply you with an example of what I think is a good annotated bibliography. I would suggest, though, try to go for more than just the minimum. March 26th will be the rough draft of your manuscript. So I will read it, I'll give you feedback. April 16th will be the manuscript final draft. And then we're going to select, we have group student presentations of the individual work. And they're in our syllabus, April 28th to May 5th. So I need your also to rank in terms of preference, which days you will be like to present your work. Okay, so you're going to give me 2 things. You're going to give me eventually the selection of topic based on preference, and then we're going to decide on when you're going to present your actual work. Although roughly it's the end of the semester, so no one's going to be disadvantaged by this. All right, so that's basically the discussion I wanted to have to give you the context about the project. If you have any questions or doubts, please let me know as soon as possible, because we do have the upcoming deadline for the selection of topics, which will be based on your preference. And so I'll help you to decide what ultimately will be what you will focus on. All right, everyone. Thank you. And I'll speak to you later.